First of all, thank you, Gabs, and thank you, Lucia, for inviting me to this seminar and, well, give me this opportunity to talk. So I'm going to present you the proximity ligation assay, especially on the context of probing GPCR dimers. So yeah, as Lucia mentioned, I'm Etienne Billard, a postdoc at Hébert. A bit about me. So I'm going to move. I come from France. I come from Morvan, so that's a natural park, quite beautiful. It had been last month said by Le Monde, top French journal, that said it was the place to be in France. So as I say, if you like lush nature and highly landscapes, you know, it's a nice place and it's very tranquil. So I did my undergrad and graduate study in Clermont-Ferrand, here on the map. Well, small but quite nice city built with volcanic rocks. It's quite cool. And so I come from a chemistry background. I did chemistry and a bit of biology in bachelor, and I did drug sciences. So it's a mix of brute organic chemistry and pharmacology. It was interesting. And then I immigrated in Canada to do my PhD with David Chatney in Laval. The campus where I was like actually quite nice by the Des Prairies River. And I was doing like the synthesis. So I was, I'm a chemist, so I synthesized libraries of peptide and characterized biased agonist and allosteric modulator of the urotensinergic system. That's the GPCR system. And from 2019 up to now, I'm doing a postdoc at McGill with Hébert, and my project deal with 5-HT2A receptor and AMPA receptor, hetero-receptor complex, so dimerization, as a potential mediator for the action of the LSD, the psychoactive drug. So why I'm interested into LSD and psychedelics? First of all, because tripping is fun, but also because, well, tripping is useful, in fact. In the past 10 years, we have what is called the psychedelic renaissance, where we see a boom into the clinical trials for what is called psychedelic-assisted psychotherapy. The idea is to use the altered state of consciousness that's happening during the trip to integrate it into psychotherapy, to bring positive outcome and change into the patient. So it has been successfully applied for the treatment of treatment-resistant depression, of anxiety, and substance use disorder. So at this point, we know they function. We don't really know what's the exact mechanism by which they function, especially the very molecular basis of it. We know that all psychedelics, LSD, psilocin, DOI, they're all agonists at 5-HT2A receptor, and that in terms of brain NMR imaging, that during the trip you can see a huge enhancement between in the connectivity between different brain network that normally does not communicate with each other, which can explain the increase in creativity, for instance. And so why it does that? It is believed to be because psychedelics stimulate the pyramidal neuron in the prefrontal cortex, in the layer 5 of prefrontal cortex. And they do so either by stimulating directly the 5-HT2A receptor or indirectly upon the presynaptic release of glutamate. And So glutamate activates diverse receptor, and we're interested in AMPAR because AMPAR, so it's an ion channel that's super important in the synaptic plasticity. And AMPAR is crucial for the pharmacology of LSD, as it is shown to mediate the prosocial behavior induced by repeated LSD administration, so that's in mice, and also the neuroplasticity triggered by LSD needs AMPAR. And what makes this project start is the realization that, oh, there are GPCR, the beta-2 adrenergic, the dopaminergic D2, that are known to form complex with AMPAR. And also that 5-HT2A, we know that the postsynaptic 5-HT2A, so the one that's expressed in the same cells as AMPAR, is able to regulate the fate of AMPAR. So the general hypothesis was like, okay, probably those two receptors might interact, form a dimer together. And so we want to know if it's true and if this complex plays a role into the pharmacology of LSD. So I'm talking about GPCR, just to be sure that everyone knows what we're talking about. So here is a GPCR. We all know and love GPCRs. They're composed by 7 transmembrane hydrophobic domain that helices that insert into the membrane, and you have on the extracellular face the ligand binding domain, and upon the binding of the ligand, you have like a shape shifting of the GPCR that will allow signaling in the intracellular phase. So you know a bit about the way they function. They are GPCR for G protein coupled receptor. So upon like fixation of the agonist, you will have a dissociation of the alpha and beta gamma subunit of the G protein that will each be able to activate a subset of effectors. And you have also the beta-arrestin 1 and 2, that's important in GPCR in the sense that first, they desensitize the G protein signalization, but also they're able to internalize and also to trigger their own signaling mechanisms. Another little thing about GPCR. So for a long time, it was believed that it was kind of a switch. You stimulate with the agonist, and you have activation of the effector. And nowadays, we know that a certain ligand within the same receptor is able to trigger a subset of effector, whereas another ligand can, potentially activate more beta-arrestin versus G protein. So that's what we called biased signaling or functional selectivity, just for your personal identification. And the big stuff about GPCR is that for 30 years, it was also believed rock hard that they were single entities, just one receptor. And there's like now a huge literature saying that no, actually, they seem to be able to exist in dimer, which means so to associate together and to allosterically communicate between one another. And what the implication of that is that, for instance, compared with a single monomer that has like, different affinity and activity, in the presence of dimer, the other partner can modulate the affinity for certain ligand and not for other. It can also modulate the activity, so the potency and the efficacy. Certain ligand can signal, so to bias the signal, if you want. And also in terms of architecture, so it's like case by case, but in certain contexts, there's like one receptor that's used to signal, that's coupled with the effector, whereas the other one is not. It's just there to regulate. So basically, they work in tandem and they're doing team job. And in the case of my project, why is it important to prove the dimerization of it is because like potentially the two together behave differently at like one separately. For instance, AMPA might bias 5-HT2A signaling. 5-HT2A might also modify AMPA channel properties, so which might explain how it has such an impact in the neuroplasticity. And also it might increase AMPA residence time, it like protect it from being, recycled. But also we know that from literature that it can promote AMPAR internalization. So yeah, they're working together and they're putting their fate into the end of one another. And if we're able to prove that they interact, that would be a great advance in our comprehension of the mechanism of action of psychedelics. And also we would identify a novel therapeutic target for diverse like mental diseases. So what's the objective of our project? That's kind of the outline. Of course, we're gonna focus on this one today with the PLA to demonstrate the physical interaction between the two receptors. But also, yeah, I want to characterize the unique signaling fingerprints, like what happened at this dimer, and to be able to physically disrupt this hetero-receptor complex. I'll talk about that briefly at the end. There's like plethora of different techniques used to show GPCR dimerization that have been used. Each has their pros and cons. I'm gonna just go over the basic. You have of course, the co-immunoprecipitation. That's a good one, and that's the very, old school technique. And so if you're able to co-immunoprecipitate one receptor with the other one, yeah, it's like really they do interact. However, well, first you need antibodies, good antibodies for each of your receptor. That's a big problem. Antibodies in general are not that good and way worse than what the antibody dealer claims. And for GPCR, it's actually usually disastrous for several reasons. We can talk about it in the question time. Also for co-IP, you need to have a lot of protein to do it. So if you want to work in certain brain part, good luck. And also you work in like that's the biggest technical difficulty. You need to solubilize your proteins. Solubilize with a detergent that is strong enough to solubilize, but not harsh enough to dissociate your two proteins. So yeah, that's a funny one. You have so Fred and Brett experiment that were very, classical, that very trendy in the 2000s. So basically, you put Brett or Fred donor by like cloning in each of the receptor, and if you're allowed, if you're seeing Brett, then that means that they do dimerize. So I put easy between brackets because that's what Hébert, the undergrad student working with me, is dealing with, and he does not have a lot of fun with it. So, it's not that easy. A big problem of that is that, yeah, you enter them by cloning, which means that you cannot do those experiments into primary cells or into tissues unless you do, transgenic stuff with CRISPR or stuff. But no, like the problem, the huge problem with dimer, let me put it, is that, of course, if you overexpress the receptor together, you're going to overcrowd the membrane and you're going to see dimers, but what does it mean? Whereas if you look at in a tissue where they're not, where they have a normal pattern of expression, then dimers tend to be harder to see. And that's where, PLA has proven useful. So yeah, let's have a look at that. Our PLA, it's cool. Like it's a very old technique. In fact, in 2002. So I don't know, maybe some of you were not born. And what's cool, so yeah, zeptomol. I've never seen this term. So 10 minus 21. It's the power of detection of this method. It can like really detect single molecules. That's one big strength of PLA. You see a PLA puncta. I'm going to talk about it later. That means that you have like one molecule. And a bit about its history. So it's patented and it's a proprietary technology marketed as Duolink that have been bought by Sigma-Aldrich. That means that if you want to do PLA in your lab, unless you do, you find a way to do it DIY, you have to pass by Sigma-Aldrich, and it's quite costly. So an overview of this technique. So it's kind of, it's an immunofluorescence technique. So basically you use two primary antibodies to recognize each receptor. And the difference with regular immunofluorescence is that whereas in IF you use like fluorescent conjugated secondary antibody, here you use what's called PLA probe. PLA probe, it's two pieces of DNA, plus and minus, that are complementary. So if they are close enough together, they can hybridize. So like the next step is a treatment with a ligase and with a polymerase that will do a rolling circle amplification and will incorporate fluorescent nucleotide. So what's cool is that, yeah, from a single molecule or a single interaction, you're gonna have an accumulation, an amplification of the signal to be able to see in microscopy those big or small red dots that are like one single interaction each. Versus a negative cell. So a bit of technical consideration to set up that. Like immunofluorescence, when you do double marking, you need antibodies coming from two different species, because your PLA probe, they're going to recognize anti-mouse, anti-rabbit, each of the two antibodies. And they have to be from a species different than the tissue it's coming from. So in my case, I work with primary neurons from rats. So I use a mouse anti-AMPAR and a mouse anti-Fabry-Fitch. Bon, if you cannot go over that and you have like two antibodies from the same species and you have no choice, Sigma cells also kit to make your own PLA probe, so you can conjugate PLA probe to your primary antibody and do direct PLA. Of course, like all immunofluorescent techniques, you need good antibodies. That's the sad part of it. If you have bad antibodies, then you're going to see a huge amount of background. And so yeah, and it's quite a versatile technique. You can use it in 96 well plate. You can use it on microscope slides. So you just have to draw with a hydrophobic pen a little well around your tissue. So yeah, on cells, on tissue. Well, so I'm telling you about the protein interaction, but there's also other fun use of PLA. One is you can look at protein modification. So if you have, for instance, an antibody, So you target that protein, and if you have another antibody that's specific for, phosphorylation, for instance, you can quantify the state of phosphorylation of each of your protein or receptor. Or if you have, a nanobody that's conformation specific, you can really quantify, okay, how much of my protein are in this conformational state. That's quite powerful. I haven't tried yet. And also you can use it to do protein expression and quantification. That's the way that's been designed at the beginning. Here is a comparison between, immunofluorescence, where you have a lot of background, and that if you do PLA, where you target with its probe a single protein. In terms of workflow, seriously, it's nothing very difficult. Basically, if you know how to do immunofluorescence, you know how to do PLA. It's the same routine and about the same troubleshooting. So it's rather long, like you would work, a bit in the afternoon, and then, most of the day after. But, it's a lot of incubation steps. So, 1 hour, half an hour and 40 minutes. So yeah, you can set it up and do something on the side. But something like very important, me, for instance, I had worked with Excel that's not very well adherent. And in PLA, there's like a lot more washes than in regular immunofluorescence. So yeah, pay attention with the washes, because you might just end up at the end of the experiment and all the cells are gone. And then acquisition. So you use a microscope, ideally a confocal microscope. Confocal might allow you to really, because the PLA puncta, they're very common. They're very tiny. So with a confocal microscope, you're able to capture every single PLA puncta. And of course, like in IF, you need a negative control and your experiment, or a positive control. And at the beginning of the acquisition, you look at, okay, here is my negative control, I should see nothing. Here is my condition, I should see things. So you decide the time of acquisition, filter, height, and stuff like that, and then you read all your well with this protocol. And then with the analysis, there are numerous programs that allows you to analyze your image and quantify the dot. So I personally use Columbus, but it exists available free softwares. There's a blob finder specifically to find little dots. ImageG does that. You have also proprietary, well, you can use whatever you want. And the way that I do my analysis is that I count, I write a little routine to underline each positive cells, GFP anyway, and to count the number of PLA puncta that are distributed into this parameter. And then I express the result as number of PLA puncta per cells. So yeah, of course, you need to set up appropriate control. So the more drastic one is the one where you omit one of the primary antibody. So in this one, you should see, almost no PLA puncta. In fact, you're seeing a few per, 10 or 20 cells. And then yeah, so in my sense, I think this one is not a super useful control. It's more like it will tell you if the PLA probe cross-react with the tissue, because you should not see any PLA puncta. Yeah, a bit more robust control is one where you omit, where there's one target that is missing. And basically, to prove that you have an interaction, you always work by comparing the number of PLA puncta obtained in your condition versus the negative control. So this control, where you don't have one of the target, it's very easy to set up in transfected cells, because in fact, you just don't transfect it with that partner. So yeah, a bit more, this control tells you if your primary antibodies are actually very specific. But when you're working with primary cells or with tissue, it's a bit more annoying to set up. So you can do a genetic deletion or inhibition. So siRNA, you can use CRISPR to delete one receptor. Some stuff that some authors do is to compare the PLA result between two tissue. For instance, let's say, okay, I'm studying two proteins, one in the cortex, one in the hippocampus, and in the hippocampus, one of the protein is not expressed. So we should see a lot of PLA puncta in the first tissue and not in the second one, if they dimerize. I tend to dislike this this idea of comparing with another tissue, because if your primary antibody is not specific and it cross-reacts with a protein that is present in the first tissue and that is absent in the second tissue, then you will see a big change in PLA signals, but it doesn't come from dimer, but from cross-reaction that is not present. One very good control, but not necessarily easy to set up, is if you have, a molecule. So I saw it on a paper. They do it with an antipsychotic, and they have shown by other methods that it tends to disrupt the binding, the dimer between 5-HT2A and 5-HT1A. So they just, treated with this, with clozapine, I think, and they were like, in the same tissue, we saw a difference into the number of dimer, effectively. So yeah, I'm gonna tell you a bit about my experiment if you want to set up the PLA. So first of all, I started by doing the co-immunoprecipitation in egg cells. That was like the trusted method in the lab. So I was working in egg cells, and what's cool with egg is that you can transfect it with anything you want. So I told you that, antibodies against receptor are quite bad, but here I was using tagged version of this receptor and using antibody against the tag, which, most of them are like good. So voila, yeah, I arrived at the postdoc and I had actually no clue what I was doing on. I was, just knowing co-immunoprecipitation vaguely on paper, but then, yeah. But at some point, it functioned. I was able to have the two receptor co-immunoprecipitate together. So then, boom, it was encouraging to move towards the next step, because PLA was not used in my lab. And that's something that Terry, when he hired me, really made clear. I want you to bring PLA into this lab. So of course, to do PLA, I had to control that my antibodies against MYC and against HA, the tag, were specific. So I transfected egg cell with like nothing, or with like the receptors. And so, boom, there's not a very good transfection efficacy on the slide. I just ameliorate my game a bit since, but yeah, but they are specific. That's what matter. Interesting tip in our case, we were not, yeah, we were setting up the PLA. We're kind of careful with its function, so we included the positive control D2, the D2 GPCR dopamine that is known to form dimer with 5-HT2A. So we used it as a positive control for the PLA. And so yeah, once we have that, we're like, okay, we have good antibody condition, and we bought the PLA kit, and I did it in Excels. So in this case, I transfected all my cells with GFP that I was using as a transfection control. So I was just assessing the GFP positive cells, and I was comparing for conditions. So no transfection of either receptor, and the transfection of both receptors. So what I was seeing is that with either receptor, there was no very difference between the untransfected. So that my antibody were not dirty, and by opposition with the transfection of both receptor, there was like a very significant elevation in the PLA signal. I mean the image like talk if you compare the co-expression, very strong PLA signal at the membrane, whereas like just a few dots in the non-transfected. So at this point, world was great. I had shown the presence of an oligomer between AMPA receptor and 5-HT2A in transfected ACT293 cells. So I had shown this association with co-immunoprecipitation and proximity ligation assay. And I had shown also that AMPA was able to modulate the signaling of 5-HT2A, just not shown here. However, Joy's score is short because there's two harsh words here, transfected and ACT293. So ACT293, they are far away from being a representative model when you study what are the effects of LSD into the brain. So yeah, what about in neurons now? So neurons, unlike egg cells, they're kind of pain in the butt. You need to, well, of course, dissect and cultivate them. You have to make sure that your antibody is specific, because now you're using wild type antibodies, not tagged version. They are difficult to transfect, and also you need to have appropriate control for the proximity ligation assay, because now you cannot say I'm just going to not transfect the partner. Anyway, so my general strategy was to use a siRNA against 5-HT2A and to compare if we are seeing a difference in the number of PLA puncta in the control siRNA versus 5-HT2A siRNA knockdown condition, which would mean that, okay, the PLA signal that we are seeing in that condition actually are dimers. So, okay, that's the gore part about the extraction. We don't talk about that. About the transfection, I spent months trying to transfect my neuron with lipofectamine 2000. It just did not function. So some people, I met some people in McGill that are able to, and good for them. I had not been able. So now I use nucleofection, thanks to the Professor Pastor that allowed me to use it. And yeah, when I tried it, and for the first time, one day after, I look at the neuron in the microscope, and I saw them. Okay, they were green, they were transfected. I was like, oh shit, cool. So then I was able to transfect them. So, but what about the siRNA? Because, you know, similar to the antibody, siRNA sellers, they claim that totally silence the target, but until you have the siRNA in your hand and you try it, you can get surprises. So the way, so you can validate the efficiency of knockdown. Ideally, you do it with a Western blot to show that, okay, you're lacking the protein. In my case, because there was, whoops, sorry. Because I was working with, you know, not a lot of tissue, then I use RT-qPCR to quantify the knocking down. So the way you do it, you compare the gene you knock down with a housekeeping gene, and you compare it with the control condition. And to be sure, I included as a positive control the G-beta-1 siRNA that was used in the lab now used for a decade. That's a good siRNA. And I realized that, okay, I was having knockdown efficiency. So for G-beta-1 of 70%, and for 5-HT2A about 30%. So it's not great, but good enough. Then I needed to make sure that the antibodies that I order working on the 5-HT2A and AMPA, the wild type, were good. So it's the case for the mouse anti-GLUA2. You know, it's actually super impressive. Like you have like a very clean marking on the membrane, and well, it's just a picture. But when I play with the microscope setting, I'm like, yeah, there's absolutely no marking in the control condition. And by Western blot, it's like on non-transfected egg, you don't see anything. In transfected cells, you see things. So good antibody. However, the rabbit 5-HT2A, quite dirty, very dirty actually. And in Western blot, I realized, okay, there's like a cross reaction. There's a protein in the non-transfected that that my antibodies see. So bon, it's not great, but we are able to see a difference between the transfected and non-transfected. So like, okay, let's actually give a try with PLA in transfected egg cells, and I was able to see a difference between. So it's just n=1. This one between the co-transfected condition and between the condition where I don't express 5-HT2A. So that's the PLA puncta from this dirty cross reaction was. This is actually the diagram. Like, cool, let's set it up in neurons. So the way I did it in neurons with I was co-transfecting, electroporating the siRNA along with a GFP. And so same strategy than with egg cells. I was like considering only the transfected neurons and well, counting the PLA puncta. And so control siRNA, 5-HT2A siRNA, I saw a difference. So it's just done 2 times. That is, not great. It's not day and night, but what's cool is that, okay, I have like 30% decrease of like 30% knockdown efficiency, and I have about 30% decrease in PLA puncta. So that's kind of encouraging, but I still need to work on it. And so yeah, like one type of control was to use siRNA. Another type of control, I put it into brackets, is to use, so talk about it. If we consider the interaction between these 2 proteins, if they interact physically, it's because there's an interaction interface. So there's like the transmembrane domain are important and have like key molecular signature that allow them to bound together. And the strategy we are using currently, I went back at my previous, like my PhD supervisor, to synthesize the transmembrane domain. So to synthesize a 20 amino acid length transmembrane domain. And the idea is that if we treat our cells with those TM, they will be able to compete into this interface and to break the dimer. And the strategy would be, yeah, to use like the scramble transmembrane peptide versus one of the peptide that is actually important into the interface. And we should see a difference in the PLA. And we would like do that without, you know, doing painful siRNA, etc. So, bon, of course, that so this transmembrane, I just have one at this point, but I plan to synthesize all of those, all of the TM, and to try them. Of course, I put control into the bracket because it's not, you know, it's going to be a cool control for the PLA. But once we know that we have peptide that are able to disrupt the dimer, then it's a lot of opportunities that open to us because we'll be able to break this dimer. We will be able to really interrogate what is the importance of this dimer into the pharmacology of LSD. And like the final step that I hope to do at the end of my postdoc would be to like treat animals, living animal with those TM peptide, and to be able to assess the behavioral difference with treated with LSD if this dimer is present or is broken. Yeah, well, I'm French, so I like to badmouth. So here are like two aspects of the PLA. First of all, you know, if we read this sentence, when in close proximity, so less than 40 nanometer. I remember in the beginning of my postdoc, I looked at this very picture and I was like, okay, you know, 40 nanometer. If we look at the size of AMPA, it's 55 angstrom, so 5.5 nanometer. So actually, the PLA signal I'm seeing, you have AMPA there, and you might have 5-HT2A there. So PLA might not actually show you actual dimers. Another little problem, so like figure out you're seeing the membrane from the top, and you see like a single receptor freely moving at the speed of, you know, a dozen nanometer per second. You know, I was too lazy to make an animation, but you know, you can picture that there's like bouncing, and then up, you fix the cell. So you take a snapshot. And what happened is that some of those proteins that are not interacting, that are just bystander, will be captured as dimers by the proximity ligation assay. So yeah, it raised the question if PLA characterize actually true dimers. So I personally think that this method is great. But the thing is, like, about GPCR interaction, you know, in the late 1990s, it was just totally revolutionary, and eventually some people were believing totally stupid. So like my boss Terry was one of the first one to show that no, actually GPCR might form dimers. He claims he's the first, I don't know, but Then in the 2000, it became like super trendy. GPCR dimerization, that had been a boom of paper, hundred of paper being like, oh, we show dimerization between this receptor and this receptor. And then nowadays it's not really trendy anymore because, well, first, there have been a lot of paper, but also because there's not the killer technology that allows to be like, okay, what we're seeing is a dimer and we're sure it is. So every dimerization paper, we have to, use different technique together to kind of gather, a bunch of evidences to offer and be like, hey, might be a dimer. So yeah, to finish, pros and cons. So PLA, it has the ability to detect to single molecule detection. That's very, very powerful, and to do it like quite simply. So that's tough. And yeah, it's easy to set up in terms of the actual experiment. To do IF, to do PLA. And the very big strength compared with a lot of techniques used to demonstrate GPCR dimerization, it's that it can be used in native tissue. You don't have to tweak the receptor. You're looking at intact tissue. So it's really, okay, you're visualizing your dimer. And for the cons, it's not a live cell technique. It works on fixed tissue. So you're missing a lot about, kinetic. You miss a lot. It's costly. It's super costly. It's maybe $10 per well. So if you're lucky, money will not be a problem in your life and in your research, but that's costly. And as I show you, it doesn't necessarily highlight the true dimers, but it's like, let's say consensually that it highlights at least interacted proteins. And of course, you still need to have good antibodies if you want to set up this technique. So yeah, thank you for your attention. Thank you, CAPS. Feel free to reach out to me. I'm Etienne. I'm a decent human being. I'm at the 13th floor of the McIntyre, and you can send me an email if you have any question about proximity ligation assay, about or the Morvan or anything. Thank you.
Thank you so much, Etienne. So now I guess we have about 10 minutes for questions. If anyone has a question, you can put it in the chat or put your hand up with the raise hand function. And I see Braden already has a question, so you can go ahead.
Well, first and foremost, fantastic talk. Thank you so much for that. I had a question, actually. You talked a little bit about one of the cons of it being too close together and some of the problems that are associated with that. And I wanted to get your thoughts on kind of the other side of the issue. What happens if you're working with a rather large protein complex and your antibodies are too far apart? Is there a maximum distance that they can be before the ligation fails?
So it's 40 nanometer. I think it sees something in the range of 5 or 10 nanometer to 40. But it's not about the protein, it's about the epitopes that the antibodies are targeting.
Gotcha. Okay, perfect. Thank you.
I have a quick question as well. So when you do your transfection, do you have to have an internal control to see how many copies are actually taken up by the cell, so that when you count the puncta, you know that it's like consistent across cells, or there's differences across cells because of the actual number of puncta and not the uptake of the copy number, if that makes sense?
Yeah, so this, honestly, Lucia, I don't know, because I was just using PLA kind of superficially and not very to the precision of like counting each protein. Of course, if you want to count, then you might want to use a very short time of exposure because you've seen it in my data as well as other published. It's not like single puncta. Sometimes you have like big potatoes. So of course you need to have for sure a negative control, because you will see PLA puncta a few, but no, for your specific question, I don't know. Maybe you want to at least compare with if you have another method to quantify at the beginning of experiment, to be able to, okay, here is a comparison between the two methods, and then you do the rest of the experiment with the PLA. But also, you know, that's 20 years. That's it's there, so it's a trusted technique.
Okay, thank you. Yeah, it makes sense. Michelle.
Thank you, Etienne, for your presentation. You mentioned that PLAs are very sensitive. They can detect like very few interactions. Is there an upper limit of detection? Like, do you ever get an oversaturation of signal?
Oh, it's if you have an oversaturation, then it just doesn't look aesthetic, and you cannot do quantification. It's just you're gonna see a big patch of your cell that are all red, or whatever the color. So yeah, if you saturate, then you cannot do quantification. So you can play with the antibody primary and the PLA probe concentration, and play also with the exposition of the microscope too.
Okay. So if you're ever working with, if you're looking at a membrane protein that is very highly expressed, then PLA probably would not be such a good method.
But it depends what you want to do, I would say. You know, if it's very expressed and you have a good antibody, maybe just IF is the answer. But if you want to do quantification, like for instance, I don't know, I have this very expressed protein and it can be degraded or transformed. Yeah, then I would say maybe you play. I mean, the problem with putting a lower amount of antibody is that you're gonna not see all of those. Yeah, I don't know. Yeah, I would say again, it matters really for quantification. Okay, and you might be able to troubleshoot.
Thanks, Michelle. And I see one more question from Sarah. Sorry if I mispronounced that.
No, it's great. Thank you. Well done. It was a really good talk. I learned a lot. Just a point of clarification. So in your experiments, you are using the neuronal cultures just to give you the ability to do your control experiments because you want to do a siRNA control. Well, basically, what I want to know is that why aren't you doing it in sections, in tissue, rather than culturing it, nucleoaffecting them, and then doing all of this?
Because in a tissue, it's possible to use a siRNA in a tissue. Some people do that. If you know how to do it, come chat with me.
No, I understand that.
I would be happy to.
No. I would understand that like doing siRNA would be a painful experiment to do in that case. But so I got it right that the control is the reason that you came to the pain of culture and everything. So now I wanted to ask, like, in terms of control, and
I thought also another thing with the neurons. We want to show that there's dimer in neurons, because after we're going to do the biochemical characterization, all the signaling stuff, we're going to do it in neurons.
In the neurons,
but
okay.
Ultimately, we might do that on the living organism with fiber photometry, but you know, step by step.
No, I agree. And so now in terms of controls, because you said that the siRNA is not the most efficient either. Like, but you do like, and what you need is only seeing that there is a decrease, and that would show you that the control is working. So now what I wanted to know is that you said in one of the parts that conjugating the secondary antibody to the primary antibody is an option. So now what I wanted to know is that if that is a simple thing, which I think it's relatively easy, not like as hard as doing the whole sRNA stuff. Could you use a primary, let's say a conjugated primary antibody for your 5-HT, and then conjugated antibody with your with the what is the other one for you?
AMPA.
AMPA, exactly. And then let's say that as your control, you dilute one of these with a non-conjugated antibody. So basically, you increase the competition between a non-conjugated and a conjugated antibody. If your antibodies are specific enough, then you could use this, and a lowering would mean that basically you are competing at the site. Would it be
the prime comes in the assumption that they are specific, and that's something I don't know in neurons.
Okay.
I show that, but
and then so for the neuron part, you could use always a GFP expressing like a transgenic neuronal population, so that the GFP, the counter staining would be there anyways.
Yeah, or if I want to genetically modify neurons, I can just put a tag onto onto the receptor. But the deriving neurons from iPSC, that's something we don't know how to do in our lab. And
and it's not that reliable either. No, I can see what you're. One last quick question at the beginning about the technique. You mentioned at the beginning that you can use PLA for protein expression as well. So in that case, you would use two antibodies against the same protein. Is that what you would? Okay.
Yes. So there's two ways to do it. I don't really know about that, but either you have two primary antibodies, two primary and two secondary, or only one antibody and the PLA that each detects this antibody.
Oh, okay.
Also, what I did not mention, and I think that's what they do when they invented PLA, they also do PLA in comment ça s'appelle, cytometry.
Okay.
So that's one thing you can do.
Perfect. Thank you very much. It was very useful.
Thank you for your question.
Thanks so much, everyone, for attending January's technical seminar series. And thank you again, Etienne, so much for such a great presentation. So we'll be having another one of these in February. Stay tuned for the email for that. And with that, have a great afternoon.
Bye, everyone.